the first 9 years. I then, my remit was over last year, and I'm delighted that Alfonso was able to take over and is now the current chair of the working group on health claims. Alfonso, do you want to introduce yourself?
Yes, thank you, Sean. And yes, I'm Alfonso Siani. I'm Director of Research at the Institute of Food Sciences of the National Research Council of Italy. And as Sean said, I have the difficult task to replace him as chair of the working group of claims at the NDA panel of EFSA. So thank you, Sean.
Thank you, Alfonso. And I'll just move on to our first slide, which is to remind people that this webinar is being recorded. If you don't agree with EFSA policy, I have no idea what EFSA policy is on this, but for other people who might be more informed than I, if you don't agree with it, please disconnect now. It's as simple as that. As well as having people from all over the world, I'm aware that 3 of my colleagues are listening in at Ulster University in Coleraine, and I'd like to welcome Catherine, Maeve, and Christina. They'll be absolutely amazed that I'm actually telling people from all over the world how to use a computer, because I'm totally useless as far as IT is concerned. But I think it's self-explanatory. And if you have problems, there are loads of people here who can help out, and just if you have problems, just use this box here. If you're, and we hope this is interactive, if you have questions, please use this particular box. And then, so the question and answers, we want you to submit questions up until 11 a.m. We'll try then to answer those questions between 11 and 11:30. But obviously, depending on the number of questions, we may not be able to answer all those questions in the allotted time. If we don't, and you have submitted a question, please use the website there on the screen to put your question in. So we don't want people frustrated that they haven't got their questions answered. These are the team behind the scenes, led by Valerio Cortu, who's the head of unit as NDA. And would like to thank 2 important people who have helped us with preparing these slides, Sylvia and Lang. The objectives of this webinar is to give practical examples wherever possible, to illustrate key scientific aspects to consider when preparing health claim applications. And these examples will focus on health claims based on the essentiality of nutrients and other claims, and I'll explain what we mean by that later. We give advice on health claims which have been evaluated. We don't try to invent health claims, and it's important that we do that, because these health claims are, actually, we learn a lot from evaluating. We keep talking about a case-to-case basis, and quite often a particular dossier and a particular health claim is quite unique, and it's important that we don't anticipate particular claims. There are questions related to legal framework which would be outside the scope of what we're talking about. We're looking at risk assessment here, not risk management. We'll publish the agenda, the presentation, the webinar recording, but we won't publish the log questions and answers. So that's the first introduction in the first 5 minutes. We now want to go forward and give practical examples on the scientific aspects to consider when preparing a health claim application. And then at the end, Alfonso will go over the key messages and close. And then that completes the webinar, and that's when we'll be working to get you the questions answered before 11:30. Okay, this is the outline of the presentation. The first point is to consider the type of claim. And when I mean consider the type of claim, I don't mean whether it's a functional claim, whether it's a children's claim, or whether it's a risk factor reduction claim. what I mean there is whether it's a claim which can be assessed or evaluated on the basis of essentiality, and anything that can't be evaluated on the basis of essentiality then is another type of claim. So there are two basic types of claims. We look what that means as far as characterization is concerned, what it means as far as formulation of the claimed effect is concerned, and then at point 4 we go on to just other claims. We hope by that stage you understand what claims are based on essentiality, and that would allow us then to go on to, which I think most of you are more interested in, the other claims rather than claims based on the essentiality of nutrients. But let's take a look at this type of claim first, claims based on the essentiality of nutrients. So only if the nutrient is required for normal body function, that is, it has an essential mechanistic role in the metabolic function, and or the ability to reverse clinical signs and symptoms of its deficiency, and it cannot be synthesized by the body or not in adequate amounts to maintain normal body function, and must be obtained from a dietary source. What we mean there is that it shouldn't be rubbed on the skin or injected or whatever. It needs to be eaten. Now, we were a bit surprised that claims based on the essentiality of nutrients seem to cause confusion amongst the stakeholders, because these sorts of concepts based on essentiality of nutrients has been around for over 100 years. It was a very exciting time in nutrition. There was a lot of discovery made, breakthroughs made on how to cure deficiency diseases, which no one had an idea what was causing these. They were killing loads of people. And since that time, finding out what to do about and how to cure deficiency diseases has saved millions of lives. And in fact, working out these has earned people multiple Nobel Prizes. So the evidence here is beyond dispute. And that means that they have to be treated quite differently, because, for example, we could never do a randomized controlled trial these days on that, because how could you put people who are deficient in a life-threatening illness, how could you put those on a randomized controlled trial? And similarly, if you put healthy people who are no longer deficient, then that breaks another tenet of nutrition. You won't expect to find any effect. So unfortunately, even though we know how to cure deficiency diseases because of war, famine, poverty, they're still around, and Europe is not immune to these deficiency diseases. So they are a special part. They have to be evaluated quite differently from other types of claims. So let's take the first example. It's vitamin C in normal collagen formation. We know what vitamin C does in normal collagen formation. It's involved in the cross-links of collagen. We know what the deficiency disease is. It's scurvy. And it's been estimated in centuries past, take the British Navy, for example, that scurvy was killing more people than enemy fire. So it is a very life-threatening disease. The symptoms are involved gum, bleeding gums, teeth falling out, etc. And therefore we can, knowing what the clinical signs are and how it's involved in metabolism, we can then, or you can as stakeholders, generate a claim based on the essentiality of vitamin C. And vitamin C in normal collagen function is one, normal collagen formation is one such claim. Vitamin D, the clinical symptoms of vitamin D deficiency are myopathy, include proximal muscle weakness, diffuse muscle pain, and gait impairment. Normally, when people think about vitamin D deficiency, be it osteomalacia or rickets, they think about skeletal problems. But there are muscle problems associated with here, and we can use that, or you can use that to generate a claim based on the essentiality of vitamin D. And vitamin D and maintenance of normal muscle function could be one. Obviously, these diseases, you can't, because of the legislation, say that it cures osteomalacia or can be used to treat osteomalacia, because the target population of any claim can never be sick people. They have to be the general healthy population. Okay, even though these claims based on essentiality are important. It's very important that they are treated differently from other claims. Not all claims associated with essential nutrients can be evaluated on the basis of their essentiality. Take claim on vitamin C in the immune system, and I'll take one claim here on vitamin D and the reduction of the risk of falling in the elderly. Let's take the vitamin C and function of the immune system first. There's no question that vitamin C is involved in the maintenance of the function of the immune system. It fits the essentiality business in that it cannot be synthesized by the body, must be obtained from a dietary source. But the outcomes used in this evaluation was infections in the common cold, not amongst the signs and symptoms of vitamin C deficiency. And the claim was assessed on duration severity of common cold symptoms during and after extreme physical exercise in randomized controlled trials. And it's important, obviously, it was ethically okay to do this particular trial because you were using doses of vitamin C beyond the DRVs, and you weren't looking at people with vitamin C deficiency. So that's a particular claim where we can use randomized controlled trials. It involves an essential nutrient, but this time vitamin C, but it's not evaluated on the basis of essentiality. And similarly, vitamin D and the reduction of the risk of falling in older people. And falling is a risk factor for bone fractures. So you could have a risk factor reduction, which involves an essential nutrient, but it goes beyond the essentiality. So we have a well-established role of vitamin D on normal muscle function, as I mentioned, but substantiation of this specific claim was based on randomized controlled trials showing effective vitamin D supplementation on reducing the risk of falling. And the conditions of use were not linked to nutrition claims, but specific for reducing the risk of falling. I should mention at this stage, and I know some that we've always had discussions on, yes. Alfonso at the panel, is how do you go about setting conditions of use for a claim based on essentiality? I think the risk managers have got around this very cleverly. They've said, if you're able to make a nutrition claim on your product or your thing, then you can make an essentiality claim on that. And that's, I think, a clever way around because we don't know the precise amounts that are needed to show an effect. And we know that if you do take this supplement and you don't have deficiency, then it's unlikely to show any effect. So it's a way around, probably an artificial way around, of getting conditions of use when the claim is based on essentiality. So it couldn't be simpler as far as characterization. If it's a claim based on essentiality, then all you have to do is to go to the regulation, see which chemical forms are present in foods or added to foods, and use that. That's all you have to do. If, on the other hand, the characterization, the claim is not on the essentiality of nutrients, then you have to take more care in the characterization of the food constituent. We, as assessors, need to know what the food is. The regulators also need to know what the food is so that they can police the particular claims. So, with very different ways of characterizing these claims. They can be on the basis of composition and characteristics. Plant sterols and stanols, for example, and LDL cholesterol, fairly straightforward. Resistant starch, that was the way that was characterized because it had an effect on postprandial blood glucose, and sugar-free gum and tooth mineralization. The manufacturing process could be one, and sometimes the manufacturing process is patentable. And here in water-soluble tomato concentrate, there was quite a sophisticated standardization procedure. And if we know the mechanism of action, sometimes we can, or you can, characterize on the basis of non-digestible carbohydrates. And there the claim was in postprandial blood glucose. There was a claim which wasn't substantiated on aqueous extract from white kidney beans, and there they were looking at alpha amylase activity. So that actual substance or food was characterized on the basis of in vitro alpha amylase. Now, we know with a lot of foods which are based on plant products, there's going to be variability in all sorts of components within that food, on the basis of seasonality or whatever, but we take shortcuts. I think it's for your benefit as people who are putting in these dossiers that you don't have to have a totally standardized. If you standardize on one particular component and tell us why you're doing that, then that can often be sufficient. Otherwise, a lot of these plant products would never be characterized. So, formulation of the claimed effect. I'll go again back to claims based on the essentiality of nutrients. Consider the mechanistic role on a body function. Then look at signs and symptoms of deficiency. And you could use either to generate your claim. Then you have to consider, of course, that the nutrient is actually an essential nutrient, so it cannot be synthesized by the body, or inadequate amounts to maintain the function. And again, must be obtained from a dietary source. It has to be a food. And we look at vitamin A, claims based on essentiality of nutrients. Maintenance of normal vision would be, and has been used on a claim based on the essentiality of vitamin A. We know it's required for the transduction of light and the neural signals. Night blindness is a symptom of vitamin A deficiency, and very unfortunately, severe vitamin A deficiency can cause blindness. Obviously, now claims, you wouldn't generate a claim saying that this can cure blindness or prevent blindness or whatever. It has to be a sort of functional claim, otherwise it falls outside the regulation. And then we establish the wording and the conditions of use. And for claims based on essentiality of nutrients, all you have to do for conditions of use, if you can make a nutrition claim, that suffices. Now, there are other aspects of vitamin A. Vitamin A is a killer, especially in children because of the defects in the immune system. So there could be, immune system could be another another claim based on essentiality of nutrients. Maintenance of bone is an interesting one because there is emerging evidence now that retinoid derivatives of vitamin A do have an effect on bone. We don't know what the precise mechanism is, so it's called emerging evidence. We wouldn't put that down as a claim based on the essentiality of nutrient A, vitamin A. And maintenance of nails would be another one which wouldn't be able to be formulated on the basis of the essentiality of vitamin A. Iodine, another interesting one. Iodine is another killer, also causes a lot of problems, especially during pregnancy. But again, this one, you could generate a functional claim by saying that it maintains normal thyroid function, an essential component of thyroid hormones. There are signs and symptoms of thyroid dysfunction, it's apparent in iodine deficiency, and the conditions of use. If you can make a nutrition claim, then that's all you need. You can make the claim on essentiality. Maintenance of hair and maintenance of nails falls outside such a claim based on essentiality. DHA is an interesting one. It's a little bit more complicated, but we have substantiated one on DHA, maintenance of normal brain function. And normal brain function is a very broad, diffuse type of term, but that's what we need to do when we're looking at claims based on essentiality of nutrients, because we recognize that deficiency symptoms, they are involved over a wide range of tissues. They have multiple effects, so we wouldn't recognize something as broad as that in other claims, because it'd be very difficult to get measures of maintenance of normal brain function. But it's okay with the essentiality of nutrients. It's a major structure component of the brain, tissue, and retina. We know what its functional effects. The problem with nutrition is that it's alpha-linoleic acid, actually, which is the essential nutrient, and we have a limited ability to synthesize DHA and EPA from ALA. The ALA deficiency has caused neurological impairment, but I think we recognize that the essentiality of ALA derives from because it's a precursor of DHA. We can establish the wording and the conditions of use, and you can submit the application. We have looked at our claims on DHA which fall outside the essentiality of nutrients, blood triglycerides. We have substantiated that one, and that isn't involved with the essentiality of DHA, and LDL oxidation, which wasn't substantiated. Now, the formulation of the claimed effect for other claims can be a little bit more complex, and I know some applicants have problems with getting especially the health relationship involved with a particular claim. The first thing to do is really to do an exploratory review of the human studies available. That gives you some idea of what they are measuring as outcome measures, what food constituents have been evaluated, and we, as Alfonso will explain later on, we would want to see that the evidence is strong and coming from quality intervention studies. Do the outcomes describe beneficial physiological effect? And again, there are a broad number. It doesn't have to be a direct effect on a health or disease process. We can have indirect effects. Are the outcome variables direct measures of the claimed effect? Are the assessment methods appropriate? So there are various ways to look at this. And again, using our examples, cocoflavanols, do an exploratory review, first of all, and this was one that was submitted to us. The outcomes assessed in human intervention studies, there were blood pressure, and that's undeniably a beneficial health aspect, whereas lowering blood pressure is beneficial almost right up to a point of fainting. Endothelium-mediated vasodilation is, again, it's easy to see that could be beneficial effect. Platelet aggregation, insulin sensitivity, very clear. Lipid peroxidation, perhaps less clear, but it's important to keep lipids from oxidizing when they shouldn't be oxidized. Cognitive function, again, a very broad terminology, but it's something that obviously has a beneficial physiological effect. The only one there that doesn't, not because it's not a beneficial physiological effect, it is, reducing the incidence of Alzheimer's disease is definitely something that I'm getting more and more interested in as I get older. But the problem is that that would fall outside the scope of the regulation, because you can't say that a food reduces the incidence of disease. And the problem with Alzheimer's is, what do you use as a risk factor? So that gives you an idea of what an exploratory review can give you. And then once you look at these studies, you'll find that most studies are on endothelium-dependent vasodilation or on reducing blood pressure. Few studies on other outcomes with mixed results. So the candidates for claims are maintenance of normal endothelium-dependent vasodilation. Instead of a functional claim, if you want to go to disease risk reduction claims on diseases for which high blood pressure is a risk factor. And that's fairly easy to, blood pressure could be a risk factor for heart disease, even for hypertension. So some of the applicants we notice have a problem with maintenance. And really maintenance is just a way of getting around the risk factor reduction, because if you say maintenance of normal endothelium-dependent vasodilation, you're you can't say the maintenance is just a terminology. The evidence we would require for both the functional claim and for the disease risk factor reduction would be exactly the same. We want to see an effect of the food in lowering. So that sometimes causes people confusion. So that's all I really want to say as far as the essentiality of nutrients, and I want to move on for the rest of this talk now on the what to do for other claims not based on the essentiality of nutrients. So to sum up, where we've got so far is that you have to make a judgment on whether you want to claim based on the essentiality of nutrients or whether you want to make a claim on another claim which is outside the scope. If it's on the essentiality of nutrients, it's a very simple procedure. You look at the physiological effects or biochemical effects or the conditions of deficiency disease, and you generate your claim. Conditions of use, very simple. The characterization, very simple. It's something that some people have great difficulty with. Is there anything you want to add that might help? I don't want to spend too much time because I think most people are not interested in essentiality. There are some people interested in essentiality, and we do always get a lot of comments. I think they're more interested in the process. They're interested in whether their food product and their particular microorganism can get this light touch. So I think the process is what they're more interested in. Now, having said that, I'm not denying, and we're not denying, that a microorganism like lactobacillus or whatever, some species for that, could have a particular important effect in metabolism. It might even have a unique aspect of metabolism. It could be that quite a number of us are suffering from lactobacillus deficiency. I don't know, but at least we would expect to give some evidence that that is happening. If people wanted to look at this light touch business, I think what we can do, people who want to get evidence from their particular microorganism, it's easy for them to do so. They can set up a randomized control trial. It's knowing what we know about the microbiome at the moment. There'll be no ethical considerations in randomizing people to so-called deficiency states of lactobacillus. Now, what we couldn't do is if we couldn't randomize people to placebo who are suffering from a life-threatening deficiency. And I think that in itself differentiates what we know about deficiency diseases and claims-based essentiality and other types of claims. It's a whole level of evidence which is beyond, and that's why we take a very light touch on the process. Do you want to add anything?
No.
Okay. So let's move then on to the comprehensive review of human studies. And before you do the comprehensive review of human studies, it's important that you take a look at the published, unpublished studies. We want to see in the dossier, you want to see that they're actually the search that you used, the decision tree that you used, why you excluded publications. You want to see that the food complies with the characterization procedures that you've set out, and the suitability of the study group. And even when you do an a priori comprehensive review, you may realize that there are only studies with a different food constituent, so you would need to refine the thing. There are only studies in unsuitable study groups, and sometimes that's a scientific question, but sometimes a risk management could be used, people who are on medications, for example. So there you could consider changing the target population or conducting additional studies. But if you have at least one study in a suitable study group, forgetting about the quality of the stage, you could proceed with the application. And I'll give you examples from the comprehensive review of human studies. The fixed combination of two microorganisms in defense against pathogens in the GI tract. Most studies are conducted with one of these organisms once you do your comprehensive review. No studies of fixed combinations, so their fairly simple remedy would be to consider reformulation of the food constituent to a single strain. Example 2, a microorganism in defense against pathogens in the GI tract for the general population. Only studies in infants, and because of the physiological aspects of the gut and infants versus adults, then extrapolation of results to adults not possible. So you could consider changing the target population and the scope of the application from a functional Article 13.5 for the general population to a children's claim, which is covered under Article 14. And the third and final example here is glucosamine and joint function. And if you only have studies in subjects with osteoarthritis, we have taken the view that the joints of these people are with osteoarthritis are different biochemically, physiologically than in the healthy joints. So what you could do is consider conducting studies in healthy study groups. So that brings us then on to the very important aspect of quality, and that's where I hand over to my co-presenter, Alfonso. Alfonso, is there anything else you would like to say before?
No, I only wish to thank you because your presentation on the review of the evidence on the study allows me to go directly to the evaluation of the quality of the individual human studies, which is the next step during the process of assessment. So it's fine with me. I can go on, and I hope to be as much as possible as clear as you.
Oh, you're too kind.
Okay, so here we can we will focus on the evaluation of the quality of the individual human studies, and I want to say that different tools, different instruments are available to evaluate the quality of the study. So the applicant are not forced to use a given tool, but instead to focus on some common core concepts that are available for assessing study quality. I wish to remind that a few months ago, EFSA published a technical report on these tools available to evaluate the quality of the human studies. But another advice is to seek the help of a biostatistician or an epidemiologist that can help you to, to give this type of an evaluation. The first thing is to evaluate the human intervention studies. Why? Because the human intervention studies are the studies really needed for the assessment of an health claim. And among the human intervention studies, the randomized controlled trials are at the top of the hierarchy of evidence. So here in this slide and the next one, we present a sort of checklist that you should have very clear in mind when you prepare your dossier for application. The first thing is the study design. It is the study design appropriate to evaluate the effect that you want to show, to demonstrate. The characteristic of participant. Are the participants selected in a target population for your claims? Because otherwise, as Sean already said, you should select a different target population if this one is not appropriate. And the setting is very important because the settings should be clearly identified and described in the application. If the setting is, let's say, an hospital, university hospital, a general practice, a community like a school, a free living population, and also the geographical setting is important because the geographical setting may influence also the outcomes. Geographical factors are associated also to other lifestyle conditions that may differ. So in general, we also evaluate studies coming from outside Europe, but in general, the result of this study should be confirmed in European populations, not a requisite, but in any case better. The intervention, both the intervention and the control food should be clearly described, what the composition, the timing of the intervention, and all the characteristics related to what you administer to the people participating to the study. You have to clearly define the outcomes. Better to define the outcome before the study, identifying what is the primary outcome and the secondary outcome if there are secondary outcomes, and then to have a precise sample size calculation based on the primary outcome. And the sample size calculation in many studies we know that is conducted after the ending of the study, but the good practice is to conduct the sample size calculation before the study. Also, randomization is very important because you have to check the eligibility of the participant of the study before randomization to avoid the surprise the people that are not eligible for the study. It happened. And also you have to describe very clearly the blinding process, if there is a single blinding, a double blinding, how performed this blinding process, and all should be in the dossier. Then a very complex and important part is the main data analysis, because
that causes a lot of discussions in the working group.
Yeah, we in some cases we are facing analyses that are not clearly described. The research hypothesis is not clearly described, and also the result we will see are not clearly presented. Also focus on additional data analysis. If you want to analyze subgroups, for instance, it could be done, but it should be pre-planned in your plan. Also important to remember, better to put into the description of your study if you plan to have an intention to treat analysis or a per protocol analysis, and not according to the result to decide after the study if it's better the one or the other, because one shows what you want and the other not, which is not good research practice, I think, and I think it should agree on this. Totally, yeah. And also the result should be completely described, clearly described, and the conclusion should be consistent with your results.
Sometimes it can be embarrassing when you've got studies published by yourself earlier where you haven't actually been as exacting as you should in the things. So it happens to all of us that even though we're evaluating,
so these are general principles, not only valid for the EFSA application, but for the good scientific practice.
And that's what I say, we do learn an awful lot from high quality dossiers once they come in when we're evaluating them. And that's, I think, very good to be a member of the working group and the panel, because you do learn a lot on how to conduct studies.
So here we have this traffic light, the green, the yellow, and the red. The green are the study at low risk of bias, study good quality, randomized controlled study, well conducted, well described. In these cases, conclusion can be drawn, negative or positive, but in any case, conclusion can be drawn about the effect of the food constituent on the outcomes. There are also studies at moderate risk of bias. Some example of moderate risk of bias could be studies in which the description of the methods is not so clear, or the description on how the outliers are considered. But in any case, the conclusions are sound enough to allow us to describe draw conclusion on the outcomes. And other studies, study at high risk of bias, we will see some example of this study to make clear what we consider high risk of bias. Starting with an example in which the problem is in the test and control foods, because the test and the control food, we know, should obviously differ with regard to the food constituent that could affect the claimed effect. But it may happen, here we have the example of dark versus white chocolate for a claim on cocoa flavanols and blood pressure. These two foods not only differ with regard to the content of flavanols, but also with the content of caffeine and theobromine that are compounds that could affect blood pressure. So in this case, the study design is not appropriate for this kind of claim. And again, other study in which the subjects are not blinded to the intervention, which in general is a problem, but is even a more important problem if the outcomes are self-reported. If you use a questionnaire, like the questionnaire that are used to assess the gastrointestinal discomfort, you cannot have the subjects not blinded to intervention, because this is not good practice.
But of course, sometimes in interventions, you can't blind
Yeah.
use of the particular food.
Yeah, but in that case, you should rely on measured outcomes and not self-reported outcomes. You are right, but measured outcomes are better in that cases. Another problem flow that we encountered is the randomization conducted prior to the eligibility check. So the subjects were excluded after the randomization because were simply not eligible to enter the study, and it caused a loss of randomization, which is a high risk of bias. And another phenomenon that could occur is the dropout rate higher in one of the two groups, in the intervention or the placebo group. That could be due to a particular characteristic, side effects, let's say, in the intervention food, and also in the case, the imbalance of the two groups does not allow to make a sound conclusion on the effect. Again, when the two groups at baseline, the intervention and the control group, differ for the outcome of interest, but also for other important variable, I would say, if the group have one of young people and the other of the older people, the two groups are not balanced. So in this case is a problem, is a failure of randomization. But in this specific example, if you have two groups, when looking at the claim on defense against pathogens in lower urinary tract, we have two groups with urinary tract infection difference at baseline, of course, disease can be considered a failure of your randomization process. Again, you designed a crossover study, and then you treat the data as a parallel study, which cannot be done, of course, because you pool together the result from the intervention and the control period. You use an inappropriate analysis for the study design. Here, the problem of multiple outcomes. If in a dossier or in a study is not clearly identified which is the primary outcome, and you have 20 outcomes that you consider relevant with only one positive, since you do not define the primary outcome a priori and not just for multiple testing, you have a high risk that this positive outcome is simply a chance finding. After the assessment of the quality of the study, another important pillar on which we base our evaluation is the biological plausibility. There are enzymes or biological compounds that can be affected by the food because they regulate the claimed effect. For instance, the hydroxymethyl glutaryl-CoA reductases is known to modulate blood cholesterol concentration. The angiotensin enzyme, angiotensin converting enzymes, modulates blood pressure. The endothelial nitric oxide synthetase and the nitrosylated species in the plasma modulates endothelium-dependent vasodilation. So if you see a variation of the expression of the enzyme or the concentration of these biological compounds, you can establish a cause-effect relationship from the intervention to the outcome. And also you can look at the compounds with known mechanism of action that are similar to the food that you are claiming the effect. We had an example with monacolin K in red yeast rice preparation. Monacolin K has a chemical structure very similar to lovastatin, which is one of the statins used to lower your blood cholesterol. So in this case, you can use this similarity for biological plausibility. But also studies in vitro are useful to assess the mechanistic, how the compound work, how our food can work. Red yeast rice preparation showed in vitro an inhibitory effect on the activity of the hydroxymethyl glutaryl-CoA reductase, and there are tripeptides in milk whose activity is similar as to that of the ACE inhibitor drugs because inhibits the angiotensin converting enzyme. And the water-soluble tomato concentrate, this sophisticated product that Sean already illustrated as, showed in vitro an inhibitory effect on platelet aggregation. But we can move from the in vitro to the bioavailability mechanistic studies in humans. The pharmacokinetics and pharmacodynamics instruments could be used to assess if the food constituents is bioavailable, that is, reaches in its active forms the target sites for the effect. We have here the example of the plasma concentration of cocoa flavanols that increased after. You can see, you administer these cocoa flavanols, and you can see these products or byproducts into the plasma. And also, if you see an effect time which is consistent with pharmacodynamics, you administer the cocoa flavanols, the plasma concentration increased, but also the vasodilation increased with a similar time course. And when the concentration disappears, also the vasodilation disappears. So there is the possibility to assess a cause effect and also the plausible mechanism of action, because if the nitroxylated species increases along with flavanols, a vasodilation is a sign of cause-effect relation, and also because this vasodilation does not occur if you first administer inhibitors of endothelial nitric oxide synthase. Again, you can use the studies of bioavailability or mechanistic studies in animals or in vitro. In vitro, you can show that the 37 compounds in the water-soluble tomato concentrate are able to inhibit platelet aggregation in these in vitro models. Platelet aggregation is very good in vitro models, quite effective to show the possible effect of the compounds. And efficacy studies in animals. You can conduct efficacy studies like those conducted in humans. You can increase your power, you can support the evidence with efficacy studies in animals. So you can administer this compound to animals and show dose-dependent inhibition on markers of platelet aggregation in animals.
So the platelet aggregation, this was one where we, as a panel, we have to look whether the wording actually is fine with the science. And I know that the applicant in this particular regard was not happy with the platelet aggregation, but then again, that is outside our scope. It's with the risk management.
At the end, to define what is the final wording for the claim, yes. So from what we said up to now, it's evident what are the criteria on which we base our scientific judgment on the efficacy, with the main evidence which comes from human intervention studies, high-quality randomized controlled trial, in suitable appropriate study groups, and then also possible human observational studies, if these studies are available.
Quite often applicants ask are unsure why randomized controlled trials, high quality, are at the very top of the hierarchy of evidence. And there's a very good reason there, is that it's important to show that the food can manipulate the effect and it's specific for the effect. So that can't be shown in the observational study. So those are the two most important. What you can show by observational studies, you could have a dose effect response. That helps as well. You could look at the consistency if you combine both the consistency of the effect. And we don't have to have it, but it's very useful to have a mechanism for the effect. What we don't do, Alfonso, is maybe worth mentioning. There is another way of looking at cause effect and evaluating, and that's the size of the effect. Now, we've had long discussions in the working group about the size of the effect. It's a very important aspect of cause and effect. We then got into the situation, do we really expect applicants in every study to show that it's clinically relevant,
because
biologically relevant?
There is a difference between the simple statistical significant effect and the biological or clinical relevant.
But if we're convinced that the study is well designed, that it's powered properly, all we would look for would be is there a statistical effect in the right direction? So we don't go in. So that's about the only traditional measure of cause and effect, or evaluation of cause and effect, that we more or less ignore. And I think that's the participants or the applicants' benefit.
So again, along with the main evidence, also supportive evidence is considered if there are human intervention studies in a group different from the target population, and also animal efficacy studies. But these type of studies alone cannot be used to form our judgment of the evidence. And as already described, the other line of evidence is the biological plausibility of the effect. So here we have some example of the evaluation process, just to make clear that different degree of evidence could lead to the same result or to different results. Here we have the example of arabinoxylan and postprandial blood glucose reduction. In the dossier was present one high-quality dose-response human intervention study, very well conducted in a suitable study group, showing a positive effect of this food on the claimed outcome. This effect was replicated in another human study that looked not at the postprandial blood glucose reduction, but a surrogate marker, like, for instance, the glucose or insulin response after glycemic load, which could be considered a surrogate marker for the effect. And also there is a well-established mechanism for the soluble fiber in reducing the absorption of nutrients. We know that soluble fiber increase the viscosity of the meal bolus, thus slowing or reducing the absorption of glucose in this case, but also the other nutrients. So combining the evidence from the randomized trial from other studies and the biological plausibility, the decision was to proceed with this claim. But in other cases, it's possible also proceed with different criteria, different type of evidence. Here we have a study on limicol and LDL cholesterol reduction. Limicol was a food composed by more than 5 different ingredients, each of them not showing an individual effect on LDL cholesterol lowering. This is really important because there were presented also studies in which each of the ingredients were evaluated, but the effect was not there. And also the dose-response relationship was not so clear, but the effect was consistently shown in several randomized controlled studies conducted by independent research groups. So even if no evidence for a plausible mechanism of action of this food was present, the consistency of the result of the randomized controlled trials was enough to allow us to make a positive judgment. Maybe in the future the idea should be to conduct also mechanistic studies.
We look at the consistency of all the totality of the evidence. Are we having effects?
Again, is a very recent dossier we evaluated during, and we had a longer discussion in the working group and in the panel too. The creatinine supplementation in combination with resistant training increased muscle strength. The dossier was quite large. A lot of studies were presented. 3 studies showed a consistent effect, 2 inconsistent effect, and other 5 showed no effect at all. But looking in detail to this study, one was short duration, In another one, creatinine was given only on training days, that is only 3 times a week. And in addition, there was a plausible mechanism of action for the creatinine administration. We know there was also another claim evaluated in the past that the creatinine administration increased the creatinine phosphate pool at the level of the muscle cell, thus improving the restoration of the ATP after the strenuous exercise. So combining the effect, the evidence from studies showing a positive effect with the biological plausible mechanism, the decision was to proceed with this claim. Another example, only one high-quality human intervention study. This is a proprietary brand of coffee C21, and reduction of DNA damage. The applicants presented one high-quality human intervention study, but not replication in other studies, and in addition, no studies showing evidence of biological plausibility. So, in this case, what the advice would be to add human evidence, other efficacy trials, and also to look at the mechanism, if there are mechanisms on this. So I think that what is really important is to consider, you have a dossier that is submitted to EFSA for the evaluation of a claim. And in this dossier, you have positive and negative studies, but to be very careful before going on with the application. If the negative studies or the not well-designed studies can affect the overall weight of evidence, because in some cases you have inconsistency in the results of the human studies presented to the panel for the evaluation. So here we have to refine the claimed effect. I don't want to spend a lot of time on this because the cocoa flavanols, but simply to say that in some cases we start with one claim and then we end up with another different candidate claim. Here, even more complicated, you have a claim in a combination of essential fatty acids on cognitive function in children, but the studies were only presented on diseased children. That is a case that cannot be evaluated, and also no biological plausible mechanism, but one study showing some effect on working memory. So the advice is to consider additional studies with targeting working memory as primary endpoint. So the wording for the essential nutrient is quite easy, Sean already showed us. For other claims, the wording is on the function plus mechanism, if possible. And the condition of use is the lower effective dose of consumption, considering also the food matrix and the food preparation for use. Consider that each claim is unique, is it considered on individual basis, and the scientific requirements have to be considered in the context of specific claim. And the very important, always look at past evaluation. The example that you could find in the past evaluation could be very useful for your dossier. So I think that we are finished now.
Right on time. Well done,
right on
Alfonso.
time. And thank you.
Okay, thanks. So that ends the webinar. And just remind people that we will try to get the questions answered within the next half hour. Okay. Bye, everyone. Thank you for your attention.